All right, so nervous system communication. Thanks for joining me today. So, with our nervous system communication business here, we are going to be talking about neurons. And neurons are the cells of the nervous system. And the nervous system communicates what we often think of as electrically. And the electricity is not the movement of electrons, but it is dealing with voltage and charges. But we develop those voltage and charges with ions, which are just elements or specific atoms that have gained or lost electrons. So we have a neuron here. And here is the dendrite side, the receiving part of the neuron. Might have a little cell body here, but then as we send the arm, this is what we call the axon. And again, it can be up to like a meter long. So the axons can be incredibly long. When we think of a nerve, we think of bundles of these axons all wound together, some going up, some going down. They can be mixed, they can be all heading in one direction towards the brain or away from the brain, but a nerve is just a bundle of axons. Here we have the synaptic or the axon terminal, the end of the axon, and that is where we have our synapse or the connection between one neuron and another neuron. And this little gap here between is called the synaptic cleft because they don't touch. And what else do we have in this drawing? Because when you have your assessment, this is what you're going to get, and you have to explain our stages of cellular communication from the nervous system standpoint. This is a receptor. These are chemical stimuli. This is going to be a sodium ion channel, as with all of the rest of these. This is a calcium ion channel. The green dots are calcium ions, where all these blue dots are sodium ions. And they're all positive cations because they have lost electrons, and now they have more protons than they do electrons. Here, this is a vesicle, and inside the vesicle are neurotransmitters. And a vesicle has a membrane around it, a lipid bilayer, and it's the same type of lipid bilayer that surrounds the entire outer cell membrane. Then on the next cell, we again start over with a receptor. But instead of like being a receptor focused on um, odor, odor chemicals, this is going to be a neurotransmitter receptor. And then that's going to hook up to a channel, and these guys work together. So this one will open based off of reception, but the other one's open because of voltage. All right, so let's move on. When we talk about our first stage, we talk about reception. The first stage is R, and at reception, we have the binding of a chemical to this protein, chemical to this protein, and that causes the opening of the first channel. The second stage we talk about is positive flooding. So R pain, RP, so reception we have here, which causes the opening. This positive flooding, we have facilitated diffusion. All of these ions are going to move into the cell where the resting potential is negative all along here. And all of these are going to flood in because there's lots of sodium ions out here. There's very few or no sodium ions inside here. So these are going to go from where they're crowded to where they're not crowded. That is going to build up an action potential. And again, I said the resting potential is all negative through here. The voltage is negative. And there are positive ions in here, though they may not be sodium. But overall, it has a negative resting potential because of a bunch of protein anions or negatively charged proteins inside. So these sodiums rush in, so there's a localized positive voltage created here. And this is our first action potential. So we have raised the voltage, and what that's going to do is all of this. It doesn't all happen at once, but it does happen very, very quickly. We talk about step 4 as being the impulse. And for step 4 here, it's just repeating steps 2 and 3 over and over and over and over. So this first action potential that we charge, then we open, because this opens only when the voltage next to it becomes positive. So this is voltage gated. It opens, it floods, and it charges. So here's our next action potential. Basically, we are just sending action potentials from one part to another all the way down the axon. So here we go. Charge, open, flood. Charge, open, flood. Charge, open, flood. And that's our step 4, the propagation of the action potential along the axon. When we hit the axon terminal, again, we've done all of this, and this action potential, instead of triggering another sodium channel, we are going to open a calcium channel. And again, the calcium is more concentrated out here, so it will diffuse in. It's going to bind to a protein that is holding these vesicles away from the outer cell membrane. When that binds, it's going to change the shape of the protein, which allows this vesicle filled with neurotransmitters. It allows the membranes to merge, which creates exocytosis, another form of membrane transport. So then the neurotransmitters diffuse across the synaptic cleft, and that is our step 5 right here. So calcium flooding, allowing the neurotransmitter membrane to fuse with the outer cell membrane, which causes the release or exocytosis of neurotransmitter chemicals across the synaptic cleft. And then, so that's all the communication within a nerve, but now we're communicating from one nerve to another nerve chemically. And we would just begin all of the steps again. So this would then be reception, positive flooding, action potential, impulse along the next axon, and then we'd have another neurotransmitter release. So here we are. Step 1 is reception. Step 2, positive flooding. 3, action potential. 4, impulse. 5, neurotransmitter release. Put them all together. We receive the stimuli. We flood in positive sodium ions. We build a localized charge of an action potential. We propagate that action potential along the entire length of the neuron by charging, opening, flooding, charging, opening, flooding, charging, opening, flooding, and that is what we call an impulse. Here, for step 5, in order to release the neurotransmitters, calcium needs to flood in. It needs to allow the vesicle-filled neurotransmitter to merge. The neurotransmitter then will go through exocytosis. It will diffuse, these molecules will diffuse across the synaptic cleft to begin communication with the next neuron. So again, I'll give you this for your assessment. You need to not necessarily draw, but you're going to not draw the entire thing, but you're going to draw the stages, you're going to label the stages, and you're going to explain the stages of nervous system communication.